Please join me in welcoming our next speaker, Barbara Wakimoto, Professor of Biology at the University of Washington in Genome Sciences. She will be giving a talk titled Bending the Rules of Organelle Biogenesis, Views from a Highly Specialized Cell. Welcome, Barbara.
Thank you, Becky. So I'm going to use my time today to tell you 3 short stories to provide evidence, I think, for this theme that I'm going to tell you about, how certain cells take the liberty of bending the rules for organelle biogenesis, at least the rules that we think are supposed to be in place for making or maintaining subcellular organelles. And I'm, like many of you in this room, incredibly awestruck by the complexity of organelles within cells. So how do cells manage to make and maintain those organelles? So my system is, all of my stories are within this process called spermiogenesis, which is actually the processes, the postmeiotic processes, that transform what look like normal average cells, although they're haploid, called spermatids, into these amazing mature gametes, sperm. And all of these structural changes, it's been characterized best in mammals, in the mouse in particular. When you go this normal-looking cell to this crazy sperm, all takes place in the absence of DNA replication, pretty much in the absence of transcription, very little transcription. So I think it depends very much on translational control during spermiogenesis, as well as protein trafficking to make this highly polarized cell. So as I said, it's been studied best in mammals, but I'm going to introduce you to my favorite organism, which is this little fly, in which spermiogenesis happens takes about half of spermatogenesis and takes about 5 days. So what do I mean by this amazing cell here? I want to just remind you of the function of this gamete, and I think this is probably, you may not have thought about this in a while, but this particular cell, this is a little cartoon of the sea urchin sperm. Just to remind you, the sperm's function is to interact with the egg. It does so in this case with the egg jelly. So cell surface receptors on the surface of the sperm activate this process of exocytosis of this amazing organelle called the acrosome. And in doing so, it actually exposes on the surface of this cell these little blue balls, which are the fusogenic proteins. So this is a really smart way of sequestering your fusogenic proteins until they're in contact with the plasma membrane of the cell that you want to fuse with. And so that acrosome is an interesting organelle. I'm also going to have us think a little bit about this nucleus here, which is highly condensed during spermiogenesis, and a short story about a very long tail, the sperm tail. So these are the different organelles, 1, 2, 3. We're going to go through some examples. So first, with respect to this condensed sperm head, what we know that happens during spermiogenesis is the round spermatid, the round nucleate spermatid transforms by reducing its volume anywhere from 200 to 20 to 200-fold into this highly condensed sperm nucleus that is associated with the loss of the canonical histones, the nucleohistones that characterize somatic cells. Proteins called transition proteins come in. About 10% maybe of the histones are retained in mammalian sperm. Transition proteins do just that. They come in, and then they're displaced by another set of proteins that are called protamines. There's only one protamine in human sperm, but there are 2 protamines, the products of duplicate genes in mammals. But that's an amazing transformation, again, of chromatin remodeling to make a sperm. Then when you extract the DNA binding proteins, you get out a population of proteins we call sperm nuclear basic proteins, or SNBPs. So if we look at the sperm SNBPs across different animals, this is work by Juan Alcio and his colleagues at the University of Victoria. Again, these histones that characterize somatic cells are shown here, H2, H3, H4, and H1. If you look across different animals, you see a wide diversity of proteins that can be extracted from mature sperm. In this case, in vertebrates, in this case salmon, where protamines were first discovered, the complement of proteins is really very simple. It's predominantly the protamine. A really interesting question is, what's the origin of these protamines? And again, Alcio and his colleagues have done a really nice comparative study to show that if you look at the protamines of vertebrates, here's a small, basic nuclear protein, it is actually evolutionarily derived from a molecule they call protamine-like. And then it, in turn, is derived from the histone protein H1. There should be blue lines here, too. So what happened in the course of evolution was histone H1 lost portions of it to give rise to the protamines. So what do we know about my favorite organism, Drosophila? It also has this dramatic transformation of a round spermatid to a needle-shaped nucleus, about a 200-fold compaction. There are also histones in spermatids that get replaced by transition proteins, and also a class of sperm nuclear basic proteins. These were discovered simply on the basis of the fact that they were products of MSTs, or male-specific transcripts. And that's how they've been characterized, just a handful in the very beginning. And they were called Drosophila protamines, or protamine-like, with the presumed origin similar to the mammalian protamines, just based on the dynamics during spermiogenesis. So what did we learn from our studies of these proteins? Our entry point was actually through forward genetics, when we had a mutation in a gene that we called deadbeat, and found out that deadbeat encoded a small nuclear sperm protein. But when we did the localization, and this is a stage we call the rocket stage, there were only several foci, as opposed to within the whole sperm chromatin, and those foci corresponded to the telomeres in Drosophila. Co-localized with the telomere capping complex precisely, it turns out that deadbeat actually requires the telomere capping complex to actually be localized to the telomere. In the absence of deadbeat, what happens is the telomere complex is lost during late spermiogenesis. And the idea is that the telomere capping complex actually requires active maintenance in spermatids and in sperm during the histone to SMBP transition. So that's a different kind of role for a sperm nuclear basic protein than others, including we, had anticipated. So what else did we learn about these proteins? The deadbeat and the telomere capping complex is retained on the mature sperm and contributed to the embryo on paternal chromosomes. So if we look at the first minutes of fertilization, the female and the male pronuclei, the products of meiosis, come together during fertilization. They're juxtaposed, and they enter the first embryonic cell cycle in synchrony, and then divide to begin then your embryonic amplification of nuclei. What we know is in normal wild-type males, in wild-type embryos with wild-type maternal moms and dads, the maternal and paternal chromosomes are synchronous at this stage. But if the sperm is donated by a deadbeat dad, then these paternal chromosomes have problems synchronizing with the maternal component. And the reason is because there's a lot of chromosome fusion and bridging happening that subsequently gives rise to other cycles of bridge breakage and fusion because of the absence of telomeres. So what conclusion from those is that the sperm from deadbeat fathers lack telomere capping complexes. Paternal chromosomes are defective, so that results in a paternal effect on early cell cycle delay. Most embryos arrest early in development, but that is one of the roles for these sperm nuclear basic proteins that we hadn't anticipated. So what we also learned about these proteins, this is the one that we are now calling MST-HMG9. It's kind of boring compared to deadbeat, but what we find is that it is a member of a gene family. That's why we had to rename it. It actually has there's actually 12 of these different proteins in Drosophila with high copy number. And what they encode actually are proteins that have a central core that's conserved. This core actually has a capacity to form alpha helices with conserved aromatic amino acids that give rise to a predicted L-shaped structure. And this structure is actually a modified version of HMG, or high mobility group proteins, which are very abundant proteins in eukaryotes that normally serve as transcription factors or as architectural proteins. So in Drosophila, what happened is an HMG protein required a male-specific transcription pattern, amplified like crazy, and is now used as sperm nuclear basic proteins. So what else do we know about these? We were surprised to find when we compare among Drosophila species that the number of these proteins are not stable. Some are lost, some are gained, some are amplified to higher copy numbers. So these are very rapidly evolving proteins in Drosophila. So our conclusions from these studies, we followed the dynamic expression of these proteins, and you can see as an example here of the spermatid nucleus undergoing nuclear transformation. You lose the histones and also modified histones. You gain the sperm nuclear basic proteins. After that, those are retained in the sperm. Another example is here. So what we know when we just compile all our data, here's an example of a subset of these proteins. Histones are lost, transition proteins come and go, and these MST-HMG proteins come in at different times, and they're localized to different regions of the genome. So it's a more complex picture than we would have had anticipated based on the protamine studies. So I already just cited these particular conclusions. What I want to say is these are diverse functions, one having a telomere capping complex function, another one providing what we think is they're not necessary for sperm nuclear condensation, but we think they're necessary to give structural support to the nuclei during the process of spermiogenesis. And I'll tell you what I mean by that. What we know about mature sperm is they form these needle-shaped nuclei, highly condensed. But when we knock out 1, 2, or 3 of these HMG proteins, this is the phenotype we get, nuclear abnormalities. And in the past, people were explaining these as failure in nuclear condensation, but we know that's not true. What we find instead is that these particular abnormalities arise after nuclear transformation is complete and during this interesting process of that's called sperm tail coiling. So spermatids develop surrounded by 2 somatic cells called the head cyst cell and the tail cyst cell. Late in spermiogenesis, the head cyst cell embeds in the terminal epithelium of the testis, anchors that cyst, anchors the heads in this area using an actin-rich cap. So here's an example of that. Here's the sperm nuclei highly condensed. Here's the actin cap that gets organized around the head cyst cell and sends projections between the sperm heads to anchor those heads in there. And if that doesn't happen, the nuclei fall apart, they get disorganized, and eventually look like this. So we think these proteins that I'm calling SMBPs are required to give rigidity to the head, not to transform it in the first place, but to give it rigidity to withstand this actin-based machine as it holds these sperm heads in place. Okay? So we think these are not required for condensation, but for giving rigidity and stability. So that's the evidence for them playing a structural role. Okay, now we've actually begun to do sperm proteomics, and it's just beginning, but what I can tell you is the nuclear complement of the sperm head in flies is a lot less complex than that of mammals. They include many MST-HMGs. There are no protamines in these insects, although there are protamines, of course, in mammals. So there's an example of these 2 different organisms or animal lineages taking advantage of 2 different types of DNA binding proteins to accomplish what needs to be accomplished during spermiogenesis. So I want to move on and tell you what we've learned about this funny little organelle called the acrosome. It is a Golgi-derived vesicle. What's really remarkable is to look at the structure of the acrosome. This is this area in gray at the very tip of the sperm head in a number of different animals. And what's striking is a tremendous species-specific variation in the morphology, the size, and we're sure the content as well of the acrosome. So one question is, how do cells, spermatids, regulate acrosome shape and size as an organelle? What we can do is we can follow this, what happens in Drosophila, because we have an acrosome membrane protein. Here it is in green. We call it sneaky. I can tell you why later. But anyway, here it is at the tip of a mature sperm in an elongated flame-like structure. We have mutants that affect acrosome biogenesis. And I want to tell you a little bit about this guy here, in which an acrosome is formed, but it's small and round. So if we look at the comparison of this protein compared to if I mark this protein called PSKL, what we found, remarkably, is just as nuclear transformation begins, there are two membrane-bound compartments, two separate membrane-bound compartments at the polar end of the spermatid nucleus. And as this nucleus elongates, so do these compartments. And remarkably, the relationship is much more complex than we thought. If you look on high-resolution microscopy, here's their interactions. And this is kind of like a paper clip that you took like a little one wire up and hooked it onto the other one. But we know that that interesting relationship is required in order to give shape to that final acrosome that we see in the fly. But remarkably, that pink compartment called PSKL is actually lost during the last stages of spermatogenesis, and what you have left is a regular acrosome. So this is a compartment that's needed for morphogenesis of another membrane-bound compartment, something we hadn't expected to see. Okay, finally, I'm going to tell you a very short story about a very long tail. And this is the sperm tail. So here's the testis of Drosophila just stained with DAPI. If I pull that little testis out, I can find the different stages of nuclear transformation and the process I told you about coiling here. What's remarkable, though, is if I label the sperm tail with a GFP protein, here's the length of the flagella. Okay? So the mature sperm heads are here, the sperm tail here. So a 2 millimeter testis has a 1.8 millimeter flagella on the sperm, which is even more amazing is the fly itself is 2 millimeters. Okay? So this is an incredible task, a very costly long flagella. And what I want to tell you is that therefore, the Drosophila has managed to introduce a new step in spermatogenesis, which is called sperm tail coiling. It must coil up those tails in order to get them packaged in a reasonable fashion for it to exit the testis. Finally, I just want to say that this is another idea of modification of what we thought is classical organelle biogenesis. As the sperm cyst elongates, they're just shown here, we expected to find intraflagella transport, which is a highly conserved way to build an axoneme in all organisms. It turns out that this fly just sort of forgot to use or just stopped using intraflagella transport. It's much too slow to get a subunit added here and traffic down to the tip. So it just left the opening here at the base of the tail to add on subunits for flagella growth. So those are three examples of what I think are organelle biogenesis that kind of like bend the rules that you would think that would occur in normal cells. And I had just 1 minute of a question as to why we would see these rules being bent so often in this process of spermiogenesis. And it could be, the implications could be that I just don't understand the rules, so I shouldn't have expected them to follow these. Or these could be like the bad boy cells of multicellular organisms. They just bend the rules. The reasons may be because the sperm has only 3 functions in life: get to the egg, penetrate through the egg membranes, activate development, and contribute a sperm nucleus. So unlike many of the cells you study that we actually expect to divide and make more cells, this particular, the fate of the sperm is really just to deliver a nucleus, then depend on the egg to provide all of its subsequent necessary functions. So I think germline is an interesting example of cell types that kind of went off the deep end in terms of the rules to make incredibly elaborate structures. Okay, so I'm going to end with a thanks for the invitation to present here, and thanks to the Allen Institute for bringing the community of cell biologists together. It's really great to see people from Children's Hospital, Fred Hutch, from your institutions, and from the U Washington. This is my group here, whose names I put on the slide. I want to end with actually thanking the fly too. So here's an example of that. I just want to end with a profound observation by Ogden Nash. You guys know who he is? Okay, here's what he said. He said, "The Lord in his wisdom made the fly, then forgot to tell us why." So I'm sure he's about ready to swap one of these things. But we should bring him back and tell him that this little guy has been really useful, I think, for informing us about cell biology, and I think some of the rules on how they can be tweaked in order to make specialized cells. So thank you for your attention.
Thank you, Barbara. And again, please raise your hand so we can pass you a microphone. Okay, go with Brock down here.
Thank you. I'm just curious to what extent data supports or speculation might support a role for suppressing hybrid fertilization as a mechanism promoting the
driving force.
elaboration of these crazy structures and rapidly evolving things.
Yeah, hugely, because reproductive proteins, like proteins in the immune system, are among the most rapidly evolving proteins in all animals. So there's huge selection, just like Alex referred to for parasites. There's a huge selection for competition of sperm to be the first one to reach the egg and to not actually interact with for cross-species fertilizations. So we think the evidence is the rapid evolution of reproductive factors.
Nice talk. Just a side question. So for the sperm, like the needle-shaped DNA, once it gets into the eggs, does it also go through morphology change before it merges with the egg's DNA? And what happened to those nuclear basic proteins during that process?
Yeah, good question. So after it gets inside this cytoplasm of the egg, and I don't want you to get too upset, but it turns out we don't think sperm-egg interaction involves a plasma membrane fusion interaction in flies, but I can talk about that later. But the sperm plasma membrane has to break down in the egg. The nucleus has to be exposed to cytoplasmic factors in the egg that allow it to decondense and replicate, and it loses the paternal chromosomes then and gains maternal histones. All within, in this organism, 20 minutes.
I was wondering have you ever tried rescuing Drosophila's knockout with one of the mammalian protamines?
We have not. I think they're really forming fundamentally different structures. But this chromatin remodeling is absolutely fascinating because the loss of the eviction of the histones, the gain of the transition proteins, the gain of protamines happens all without any DNA replication. So we know some chaperones involved, but we don't really know. And I don't think, to my knowledge, people even know how the protamine wraps around the DNA to condense it in mammals.
Okay, thank you again, Barbara.
Thank you.